Welcome to Broad Eye, the podcast that explores knowledge gaps in ophthalmology and eye care.
Welcome to another episode of the Broad Eye podcast. I'm Dr. Bruno Fernandez, and today I have the pleasure of having with us Dr. Gail Siegel, research professor at the State University of New York at Buffalo. How's it going, Dr. Siegel?
Great, Bruno. It's great to be here. Thanks for inviting me.
Pleasure's all mine and of our listeners as well. So let's start with like talking about you. So what was your background? Like what did you study? And like basically your journey that brought you to where you are today.
The journey that brought me here today. Well, I started out always interested in science. And as I grew up and got along, I got very interested in small, microscopic size objects. So I would go and, look at things under the microscope, or read as much as I could about cells. And when I got to college, it became very clear that I wanted to go into research, and especially something like cancer research. And so what I did was I went on to graduate school at Albany Medical College, and then went on to do a postdoctoral fellowship at the University of Rochester in neuroscience, and started to study retinoblastoma. And that's been like a common theme of my research, starting from my postdoctoral fellowship to today. I eventually went on to take on a faculty spot at the University at Buffalo, which is, I've now just got my 20-year pin. So I've hit 20 years at the University of Buffalo, doing research there. Thank you. On retinoblastoma and all different aspects, everything from, we did some tumor studies early on, tumor formation studies, to things like differentiating the cells, getting them to become more mature cells, looking at cancer stem cells and chemoresistance, and new drug therapies, which is what we're working on today.
Yeah, so retinoblastoma it's a quite a popular topic. Like at our podcast, we had a few guests already invited, like to talk about it. It's a topic that I'm particularly passionate about as well. So let's spend some time talking about your research. I'm particularly curious about that. So chemoresistance, like I saw that you have some research projects going on about that. Like so because just to can we bring the audience like up to speed? Like, so we use chemotherapy like to treat like some cases of retinoblastoma, but there are some cases that become resistant to it. So, so I believe your research is like how to deal with those stubborn cells. Is that it?
Yeah, so we have done some work. One of the projects where we were looking at cancer stem cells, some of those, there are little subpopulations in the tumor of these stem-like cells that have that are able to resist chemotherapy. The chemotherapy just does not work on them. And all you need is maybe one cell like that to break away and to form a new tumor somewhere else in the body. And so that can be very dangerous. So you want to get rid of any cells that are resistant to chemotherapy. So once we saw that and that was going on, we've started to do some more like personalized therapy, looking at different drug targets and trying to target cells that could be involved in tumor formation. So that's one of the things that we've been working on.
And how you personalize the treatment? You do like a biopsy, and then you study the cells from the tumor for that specific patient, and then you choose the correct therapy?
That's one approach. That will, and that will be on an individual basis. There's another thing where you can look at, let's say there's a marker, like for example, we looked at HER2. That's a breast cancer drug target, but it turns out that some retinoblastoma tumors have HER2 protein on the surface, and that the drug can bind to that and kill those cells. So we have some of those types of targets going. We're always looking for new targets. Ideally, they would hit as many patients as possible and not just one here and one there. You want to try to personalize it, but yet have it be something that can apply to a larger population than just one person. I think eventually you may be able to drill down and get, the one person that has some sort of odd mutation or odd a non-typical type of retinoblastoma, where you could find a target and make a drug target and make a drug against that target.
And there's a lot of things to look for, I guess, right? So like it helps if you know what like you're expecting to find. So when you start searching for targets, like do you prioritize the targets that we already have a drug for?
Well,
Or
that, yeah, a drug target that already exists and is known and hey, is even FDA approved, like the HER2 has drugs that are already FDA approved against it. So that's always a benefit because, you know, the drug pipeline, you always have to wait for, you know, the approvals to go through, and it's much more streamlined if you have a drug that can already be used, or at least maybe you can even make a derivative of a drug that's already being used. But we're not opposed to having a new target. And we've got some, you know, that we're working on now that, you know, they may come out and may end up being good targets, but we just don't know yet. So, you know, can continue to work on that.
and is it something still purely experimental, or do you work with like actual patients and like and clinicians, like they have a patient and they send you their cells and it's like, oh, find a target for this kid?
Right now, we're not using uh, patient samples that are coming directly from a clinic. I do have access to archival specimens that are in pathology labs. So I tend to use those. I also have a lot of retinoblastoma cells, different types of cell lines in my lab, and they're in the liquid nitrogen tanks frozen down. And those we've been studying. They're good ways of studying, and you can look at live cells versus tissue that's been preserved from a tumor from 20 years ago, for example.
And some of those you developed yourself, is that right?
There is a non-retinoblastoma
Mm-hmm.
cell that I developed called R28, and
Mm-hmm.
that's a retinal precursor type cell that does not form tumors in animals. And that one, those cells are used for sort of in vitro drug testing and gene expression studies and things like that. At the time, my goal was to make a cell that was not retinoblastoma, that was a little bit more normal. They're from the rat, and they've been commercialized. People can use them for their studies. I'm always happy whenever people are able to use those cells for different studies.
Yeah, that's cool. During my days in the lab, I remember working with like some retinoblastoma cell lines, the Wary, the
Yes.
Y79,
Yeah,
the
we've got
classic
those.
ones, right?
Yeah,
So,
exactly. Yeah, we've got those in the freezer. And you have to, I mean, you have to take care of them because, you know, during this pandemic, you know, there are a lot of supply issues, and all those things are stored in liquid nitrogen tanks, and you've got to make sure they get filled up. Those tanks are filled every month. So no matter what's going on, we had to, you know, keep the cells going and keep the nitrogen tanks filled. And fortunately, my student and I have been keeping that going this whole time.
yeah, like this is the thing that like outsiders don't realize, right? Like how sometimes it's a pretty low tech, like the job of an actual researcher, like I mean, it's just like making sure the tank is
Yes,
it's
that was
filled.
really, yeah, that was my entire concern this whole pandemic. You know, we've had to shut the lab down sometimes and, you know, turn off the equipment. But the one thing that I always had to make sure of that those tanks have to be filled up. I've got cells in there from 1967.
Yeah.
And I didn't put them in there, but you know, I
Hmm.
don't want, they're like a museum, you know, you'd lose
Yeah.
everything. So that's always my
Hmm.
concern.
So still on your research, like so
Yeah.
we've talked about like overcoming mechanisms for like chemo resistance, and you also have some research projects on understanding how the actual mechanism of metastasis occur in retinoblastoma patients, and obviously like I mean how to stop that. So any color you'd like to share on that?
Well, one thing we've looked at recently is, and I think paper came out and was it 2019 or something, where we looked at adjacent to the tumor because you wonder, you know, when you know, look at the pathology of these tumors, you see, okay, obviously there's a tumor, but what's happening next to the tumor? You know, how is it spreading? And you know, are these markers there adjacent?
Mhm.
And, you know, we've done some studies that show that the neighboring cells that you might not think are part of the tumor are not totally normal. And that's important to know because, you know, you want your treatments to reach everything and you don't want whatever is, you know, becoming or is turning into something. You want that to be stopped if you can stop it.
Yeah, it's the soil, right? So it's not only the seeds, like the soil, like it needs to be amenable for the tumor to spread and eventually evade as well.
Yeah, yeah. And it's never a good thing. Like I say, you only need a couple of cells here or there to spread. And so that's, really the reason we want to try to address the whole tumor and all the
Mhm.
cells that could possibly result in spreading it.
Cool. All right, let's lighten up the conversation a bit
Yeah,
before we lose
okay.
our audience,
We
before we
stay
get
with us,
going.
everybody.
If you start going down that rabbit hole, like, it's
Yeah. Well, yeah. The other thing is we've been slowed down. The pandemic has slowed us down a lot.
yeah. Mhm.
I mean, it's just we're hoping to get things ramped up again, but it's been a very slow progress over the last couple of years.
I can imagine.
Yeah.
So, musical microscope, like, and you do have
Yes.
quite an interesting outside hobby. You play the clarinet.
Yes, I do.
And like, how did you happen to bridge like those two things, like the clarinet and like pathology?
Yeah, yeah. So so I in my house I have one room that's devoted to music. I have a music so it was a music room.
Mhm.
And I have a clarinet group, and before the pandemic we used to rehearse in my music room, and that was all it was. When the pandemic hit, I had just done a whole bunch of experiments. I had a lot of slides to look at, pathology slides, and I had shut the lab. This is March of 2020. Lab is closed, I thought, for just a few weeks. I brought home a small microscope, not my big fancy one, just a small microscope with a little camera and the slides, and I set it up in the music room. And I was able to do some slide analysis and photography in the music room. And so I ended up calling it the musical microscope room. So then what
Mhm.
happened is, also being alone at home, I would take my clarinet out and I would try these apps where you can play along with yourself 3 or 4 parts together, and I would record them in that room. And I always make sure that the microscope is in frame so you can see the clarinet and the microscope when I'm playing. And still doing that. I post it on social media and people get a kick out of it. I call it the musical microscope room.
But you actually like look at microscopes while you're playing, like just like.
No, they're not at the same time. I usually have the microscope in the background behind
Hmm.
me, and I try to make sure it's there so people know, this is the musical microscope room, so they know.
Cool. That's, I mean, we're gonna talk about like the life of a researcher, like, I mean, what it takes. And one of the things that's super important, like, I mean, it's for people to have outside hobbies, like, and I
Yes.
think that exemplifies that.
Yeah, it's so different. It's totally different from the lab, and it's just something fun. It makes me happy and I enjoy it. And you need those things, not just during pandemic, but all the time. You have to be able to do other things.
Yeah, no, true that. For the sake of mental health, like, it's good to disconnect and have activities that are completely different from your day job.
Exactly. Yeah, I am not thinking about retinoblastoma while I'm playing music. It's a totally different thing.
Yeah. And usually that's when insights happen, right? Like
Yeah,
when
yeah. A
you
lot of
get
my
our
best
minds out of
ideas
things. Yeah.
are, yeah, I'm driving along the highway and the idea for those R28 cells came to me while I was driving along the highway going somewhere. It's, oh, what if I did this?
Yeah. And
Yeah.
and on that, so you actually have a lot of time, right? Because you seem to commute like a long way
Yes,
to
I have
work every
I
day.
have a very long commute. I live about over an hour between my house and my lab. So, and especially on a day like today when we're having a storm, it can be challenging to do that commute. But
Yeah.
fortunately, even before the pandemic, I've been doing remote work. I always try to have at least a day where I work from home, and you're always working on manuscripts and
Mhm.
grants and, you know, different things you can do from home. So thank goodness for the technology.
Yeah. Uh, all right, switching topics again. I really want to know more about another initiative that I'm involved on, and it has a quite a similar goal than to a podcast, which is like to make science more accessible. So it is the Rochester, like, and New York March for Science. Uh, I'll let you explain to us
Yeah.
like
Oh,
what
I'd
exactly
love
that
to.
is.
Yeah, I'd love to tell you about the Rochester, New York March for Science.
Uh,
So it started back in early, like February of 2017. A group of us, thought, we really need, there seems to be a lot of anti-science sentiment out there. And we would like to, bring science to the people. The more education and the more outreach that we can do, the better. So a few of us got together, and one of the founding members, we are a nonprofit, non-political organization. So, we don't endorse candidates or get into politics or anything like that. But our goal is to do scientific outreach to the community, and we're in Rochester, New York. And what we normally do is that we do different events. We had we've had a couple of really big rallies where a couple thousand people came, and we went to the street and we marched and we held signs, March for Science. We had expos where we had people demonstrations set up where people could come and look at different types of experiments and different technologies. We've had, and then we started to move. Now we're on a little bit of a smaller scale where we've gone to farmers markets. We set up a table with a demonstration. Here's a physics experiment or something like that. And it's usually kid friendly. Kids come up to it and try things out. We've gone to libraries and done, things in libraries. Once again, the pandemic has, we haven't really had many events. We did some farmers markets when the COVID numbers were a little lower, and then we had to kind of stop. But these are the things that we try to do, and just bring the science to the people and make it more accessible.
And is it like biomedical science only, or
any science? We have people doing, conservation. We have, all different types of science. We do not, limit it to any one particular type.
And those like outdoor initiatives on farmers markets or fairs, how's the reaction? Like, do people seem interested, and do they come know more about it?
Yeah, they love it. They love it. They come up, and we have a sign out, March for Science, and we have little, stickers and bumper stickers and things and posters, and they come and look at that, and then they look. A lot of the demonstrations are hands-on. So, okay, if you mix these two chemicals together, they turn a different color, something like that. Or here's a microscope. You can look at, what's in the pond water. One guy had this really cool big lens that he used to harness the sun. Then the sun on a little frying pan with unpopped popcorn in it. And he was able to pop the popcorn using the sun and a big lens. It's just kind of cool things that get people interested, and they can see, the value of science in their everyday lives.
Yeah, no, that's interesting. And I mean, I saw that one of the missions of it is to improve like inclusiveness and accessibility, like
so
careers
yes, I'm the diversity officer on the executive board of the March for Science. And yes, we always want to be inclusive. Whenever we've had an expo, we make sure that we have people, checking and make sure it's accessible to people, on wheels, or people. We had Braille guides for people. We tried to get as much as we could so that it was accessible to everyone.
That's interesting. And I guess the part of the March for Science is it's not only to get people more familiar with what science is, but also to what being a scientist entails. Like, is that right?
Right. I mean, we need examples. You want to, I mean, if you see a scientist who looks like you might feel, hey, maybe I could do that, versus in the olden days, you didn't see women as scientists, people of color as science. You just didn't see it. And it makes you think, well, maybe there's some reason why I can't do that. But really, we want to be welcoming. We want everyone to feel like they can be a part of this.
Yeah, and I guess there's still, I mean, it's changing, but there's still this stereotype of what the scientist should look like, right? And
yeah. No, I've had that experience. When I first opened my lab at Buffalo, there was a salesperson came, and he asked me, and I'm looking for the person in charge here. I said, that's me. No, I'm looking for your boss. I said, well, I'm the head of the lab here. My boss is the chairman who's downtown somewhere. But I still run into things like that once in a while.
Yeah, and I remember also when I used to be a researcher as well, I would tell people oh, you're too tan to be a researcher. I would think you're supposed to have thick glasses, white as a ghost, and just stay in a basement lab all day, you know. So
Right. Or if you're a woman, you can't wear nice fashions, or you can't look a certain way, or have tattoos, or everyone's got
Mhm.
an idea of what a scientist. But you'll see on Twitter I think there's a hashtag of this is what a scientist looks like or something.
Mm.
People put pictures of themselves to show,
Mhm.
you don't have to look a certain way or be a certain way.
And like bridging into the next topic there, is it
Yeah.
something that your memoir tries to accomplish as well? Like, I mean, your book and
Yeah, the memoir was
Mhm.
really, it's a story of just from the very beginning, all the way up through school, and some of the trials and tribulations that you have to go through in order to, be a scientist or work as a scientist. And you do run into things, I mean, the book has stories about someone trying to sabotage my
Mhm.
experiments,
Mhm.
and just all kinds of things like that. You never know what you're going to run into, and you just have to really be resilient and bounce back is what I always tell people. The students in my lab, you have a plan. You want to go from point A to point B, but it's usually not a straight line. Okay, well, I have to wait, now I have to do this differently, or this, I didn't pass this exam, or whatever it is. As long as you just kind of keep going in the right direction and moving forward,
Mhm.
you can do a lot, and overcome a lot of
Yeah.
challenges. I don't know if my postdoctoral advisor died when I was working in her lab. That was a huge challenge.
Mm.
And she was in a car accident. And
Sorry to hear that.
yeah, that was a terrible time where, you know, we had to put her poster together with a group. We pulled together, we presented her poster at the
Mhm.
neuroscience meeting, and people were finding out there that she had died. It was just really,
Mm.
it was one of those things that no one can prepare you for that.
Yeah.
And but then it makes you strong to be able to try to, to get through these things and be able to, then other things seem like small potatoes. Oh, okay, this paper wasn't accepted. Oh, okay, I'll just send it somewhere else now.
Yeah.
You just put it in perspective.
Yeah, and I guess you get used to failure also, right? Like, I mean, in a good way, you know, because
Yeah, I
like
mean, the
life in
purpose of
science,
an yeah, the purpose of an experiment is to figure out what to do the next experiment. You don't
yeah,
assume it's going to work the first time.
and like part of research, it's like to keep failing, so you tinker, right, and then eventually gets to something better, but you won't know what it's right until you discover what it's wrong, right?
Yeah, and you
And then
need a certain attitude, because I mean, I had people ask me, "Oh, your experiment didn't work. How does that set you back?" And you can't think that way. It doesn't set me back. It's setting me forward to know
yeah,
what to do next.
it's
But
like
if you have the
narrow it
other
down, right?
mindset, yeah, if you don't have the right mindset, this scientific research isn't for you if you can't
Mhm.
deal with it's not going to work every time, if you don't have the patience.
Yeah, and also like the exploratory nature of research, right? Like, I mean, to trying to figure out different ways of doing things or a different way of looking at things, and then I mean, you're bound to fail, right? If you start like doing things different than like the consensus, right? But that's also how eventually, like you're gonna find something completely new.
Right. I mean, that HER2 study, there was a paper that said retinoblastoma does not have HER2, but they'd only used one antibody to check for it. When I used different antibodies, I found it. So you can't always go by, one paper or one thing that someone says. You can challenge it, you can check it, and sometimes you might find something different.
Indeed. By the way, like, I mean, the title of Dr. Siegel's book, it's
Academania, My Life in the
uh,
Trenches of Biomedical Research.
there you go. It is available at Amazon, so you can download it to your Kindle. So it's really not that hard, like, I mean, for you to get access like to that. And if you ever think about like having a career into research, particularly like academic research, it's always good to know what it takes and the perspective from an insider. And from I haven't read the whole book yet, but like, I mean, it strikes me as a very, like light-hearted book and
Yeah,
quite easy to read, and like a
it
bit of
doesn't go
humor
into
also.
extreme detail. It's for the layperson. It was meant, more for that, like a memoir. Yeah.
Exactly.
Yeah. No, it was a very therapeutic for me to write it and just kind of get it all down. And then it turns out other
Mhm.
people were interested in reading it. So very good.
Was it your first like non-scientific writing?
First, I think it's the
Mhm.
first, well, I've done like little things like letters to the editor, and, but this is
Mhm.
the first real book
Mhm.
that I wrote and published on Amazon, and it was very exciting. I've gotten a lot of good feedback on it, and it just kind of takes you through just starting from kindergarten and the very first experiments that I
Yeah.
did on the ground, and
Mhm.
all the way through to the present time.
Mhm.
I'll have to do volume 2 next or something.
Yeah, that would be my question there. If there is any second, like a
Yeah,
main
well, I
book.
stopped at a certain point because you don't want to put things in books that might get you in trouble with where you are at the moment, kind of thing. So I
Yeah.
just stayed with things that happened in the past, and
Mhm.
I'll revisit, and when I retire, I'll have more to say maybe.
Well, we look forward for that day, I mean, I'm sure you have a lot to share.
Yeah, no, but it's good. A lot of good people out there, wonderful experiences. It's really been a great career, and I would encourage people to explore that if they're interested.
Cool. So Dr. Seigel, we are coming to an end. So is there anything that we didn't cover yet that you would like to share, like with our audience? Any
Um,
final words of advice for new
Well, I,
people
I
getting?
do want to thank
Mhm,
the sponsors, the
mhm.
people that have
Of
kept the
course.
lab going during the
Mhm.
pandemic. My lab is supported by the Hearing Health Foundation and by the Developmental Studies Hybridoma Bank, and I want to thank them. They have stood by us through the pandemic, given us time extensions so that we can continue to keep the lab going. It's been very important to have that.
Oh, that's very good to acknowledge, like the support, like research and chief.
Yeah, definitely.
Mhm. Cool. So I won't take you. I don't want to take much more of your time. Once again, like thank you very much for accepting our invitation. It was a pleasure chatting with you, and congratulations for everything you've done so far, and good luck on the projects that you continue to be involved on. We hope good things come out of this.
Thanks, Bruno. It's been fun. Thank you so much.
All right. Goodbye, everyone. To the next.
And that concludes today's episode of the Broad Eye Podcast. If you enjoyed this episode, please be sure to subscribe on your favorite podcast platform. Of course, ratings and reviews are always welcome. And you can certainly share this episode with any of your colleagues or friends who might enjoy it. Thanks for listening.